Hi, this is Khadija. So this is the second study of Model 1, and today we'll be talking about the sense of smell. So as we've stated in the previous class, we have the special senses, which includes the sense of taste, which we talked about is as study 1 in this module. Today we'll be talking about the sense of smell, and next we're going to be talking about the sight, and also later on we're going to be talking about hearing and also balance. For touch and proprioception, we're not going to be discussing it in this year. So brief anatomy on the sense of taste and smell, like we previously mentioned that this smell tends to interact or interferes with the sense of taste. It could intensify as well as inhibit the sense of taste. So this is because of several synaptic connection in the brain, and also we have some anatomical interaction. So initially from the previous class we said that the pharynx is running from the posterior aspect of the nose and oral cavity and all the way to the laryngeal level. So here we can see that the part of the pharynx that is located behind the nose is called the nasopharynx, and that's behind the mouth is oropharynx, and that's below is called the laryngopharynx. And we said that molecules, because some of the molecules that come into the oral and the nasal cavity can flow via the oropharynx to come into the flow into the nasopharynx and come into the oral cavity, thereby intensifying or increasing the sense of smell. And also, if you have a lot of fluid discharging from our nasal cavity, or the sinuses around the nasal cavity, or another fluid coming as a tears that is being drained into the nasal cavity, that is the inferior concha of the nasal cavity, which we're going to see today, is also going to have dilution of the textant in the oral cavity, thereby reducing the amount of molecules that might bind to the taste receptors, as a lot of the molecules will also dilute with the fluid coming from the nasal cavity and goes directly to the stomach without any of the textants binding to the receptors and thereby being detected. So this is what we discussed, and so we said that the flow of fluid from the nasal cavity down to the nasopharynx and then to the oral cavity and gradually and finally into the stomach is what is called the nasal drip. So the anatomy, we're first going to talk about the anatomy of the nasal cavity, then we'll talk about a little bit about the histology of the nasal cavity, and then we're going to concentrate on the nasal epithelia, which contains the nasal receptors, and thereby we're going to also talk about the efferent fibers of the nasal receptors. In this aspect is called the olfactant receptors, and then we're going to talk about the fibers, the first order, second order, other fibers going to the brain. So the anatomy of the nose proper, it's the size of the nose varies, and it also helps in anthropometric study where we can easily identify somebody. So without seeing a lot of people, when you see the shape of their nose, you can predict which part of the country or which part of the world that particular person is. Elsewhere, it also varies because of genetics, which is how we are individually made up. As well, climate, because we have a warmer area, that is why we have a bigger nose so that we can inhale a lot of air, while those people that are found in the pole, the South Pole, where it is very, very cold, they tend to have a smaller nasal opening to reduce the amount of harsh, dry, and cold air from coming into the nasal cavity. Age as well also shifts the dynamic or the shape or morphology of the nose, and as well as trauma. And a lot of time, the females tend to have smaller nose as compared to the male gender in the same community. So when we talk about the anatomy is the tip of the nose, apart from when we see we have the apex of the nose, we also have at that particular area we have the ala of the nose and also the nares where we have the opening, and then in the middle we have that column that separates the two nasal cavity, or it is also referred to as the phlegm, where when you remove the muscular structure, when you move the skin and other light structure, you're going to feel it having, it's like a perpendicular septum that separates the two nasal cavity from each other. Then we have the dorsum of the nose, which is the elongated part, and you can see it in the image, which takes up the majority of the part of our nose. And then we have the base of the nose, where we also have we have the bridge of the nose. So above that is where we have the roots of the nose, where the bones, the bony parts of the nose tends to fuse with the bony parts of our forehead. That is the frontalis bone. So the function of these the nose is generally for smell, and that is which we're going to talk about it, the sense of smell, and also it serves as a point of breathe in. So we breathe in a lot of air, and we also tense the epithelial or the histology makeup of the nasal cavity. It helps to warm the air before it goes into our own body. So it needs to increase the temperature of the air that is being breathed in, such that it is similar to the body temperature, so that it doesn't cause any changes. So the external, let's see the bony structure of or the skeletal structure of the nose. So when we talk about skeletal, we mean either the bone or the cartilage. So in this aspect, we have an upper 1/3, which is where we have the roots of the nose, and there we have the nasal bones, which connects to the frontal bone. And then we have the lower 2/3, which is mostly made of the cartilage. That is where we have the most of the bridge of the nose, and sorry, the dorsum of the nose. And then you also have the ala, or the apex of the nose, where you can actually get to appreciate the ala fibrofatty tissues on the external, while on the midline we have several bones that are fused together to give us, several cartilages. So in this program, we don't need to concentrate on the individual cartilages for our knowledge, but just know that the anterior 2/3 or lower 2/3 of the nose is mostly cartilaginous and fatty tissue. And so when we go inside the nose, when we try to have a coronal section into the nose, and then you look at the structures deeper into the nose, the ventral view is showing us where is showing us where we section the face, and then you can turn it and look at inside. So on the lateral aspect, you can see the eyeballs, and then on the midline you can see the nasal cavity, where the two cavity containing like a downwards projection of a tissue. Those downwards projection of tissue are called the nasal concha, and we're going to talk about them in a bit. But in the midline, what separates the two parts is the perpendicular septum, and we have the cartilaginous part coming from the ethmoidal bone, and then we also have a bony lower part which is made of the vomer bone. So the ethmoidal bone is the one coming from the nasal, from the frontal bone, from the upper part, while the vomer bone is coming from the roof of our hard plate, and it goes to attach itself with the hard plate. Okay, that is why you see the hard plate at the bottom aspect. So this perpendicular septum separates the nasal cavity into right and left. So when we check the lateral view, we get to appreciate the nasal cavity, which separates it, and then you can see where part of the nasal cavity goes all the way to attach to the hard palate. So that part of the nasal septum, that part of the nasal septum is on the lower aspect is called it's coming from the vomer, while the upper part is the perpendicular septal cartilage coming from the ethmoidal bone. So let's go back to the ventral view, and let's talk about those downwards projection. Those downwards projection, in this image you only see two of them on each side. So the one we get to see is the middle and also inferior nasal concha, but if you look at the superior, if you look at the upper part of the nasal cavity, you're going to see that we have another superior nasal concha, but it is smaller and shorter than the middle one, and that is why it's not very obvious. So this the downwards projection coming from the lateral aspect of the nasal cavity, they are called the nasal concha. So we have the superior, the middle, and the inferior nasal concha. So but you can see in this image, that is the ventral image, we have more of the middle and inferior. And then underneath each of the nasal concha, you have pocket looking structures. Those pocket looking structures are very important because they are as well named after the locations they are found. So if there is a space below that of the superior, it is called the superior nasal concha, and that one below or behind the middle nasal concha is called the mesial nasal meatus, and then finally the space below the inferior nasal concha is the inferior nasal meatus. So meatus is like a small cavity. So this is an image where we replace that particular this thing, and we showed you the bony image of that in a smaller version, and then you can see that you have the inferior, more obvious is the inferior nasal concha, and the space below it is called the inferior nasal meatus, and then you can see that of the middle is also very prominent. Superior is not very prominent. So in this image we also see the yellowy structures underneath the blue projections. So the new image, the upper image on the right hand side is the one that shows the nasal conchas, and the yellow colors are showing or indicating the space below the conchas. So these spaces below the concha are also named after the projection. So you have and the ones located below the superior concha is called superior meatus, and the one below middle is called the middle meatus, and the one finally the one underneath inferior concha, it is also referred to as the inferior meatus, which contains openings of the sinuses around it, and it's for the ones, especially the middle one, contains the opening of the sinuses, the nasal sinuses just surrounding the nose and that of the frontal bone. And then the inferior meatus contains the opening or the lacrimal duct, which empties the tears we drain from our eyes into that particular aspect, and we subconsciously it drains into our stomach via the nasopharynx, oropharynx, laryngopharynx, and then gradually down to our esophagus, and then goes to our stomach. All right, so you can see this is an enlarged view to show you this, and I hope you'll be able to compare these images and understand the anatomy very well. So like we said that this is the lateral view of the concha, so you can see them, and in the nasal cavity, so you can see them, they look like flap or flap over another flaps. So they are not so they are little projections of the bone, and they are covered by also the mucosal membrane, and the mucosal membrane contain histological tissues or cells, which we're going to talk about them later on. So you can see also in this lateral view you have the superior, middle, and inferior concha, which are all labeled as concha, and then from the posterior aspect you can see the nasopharynx, oropharynx, and also laryngopharynx. So like we said that, and these are just lectures to describe the conchas. We say conchas are bony projections, and they are covered, and what they do is they increase the surface area of the nasal cavity. So when you fold, it's just like when you have a long mat and you fold them, you notice that you've reduced the size of the mat to enter or occupy a very smaller space. However, the mat itself, when open, straightened, is going to be it's going to have a large surface area. So that is basically what happens with these bony projections, which are called the nasal conchas. So like we said earlier on, all this nasal cavity is also covered by mucous or olfactory. They are covered by epithelial tissues. And the epithelial tissues are also categorized in different areas, which we're going to talk about them. And these epithelial tissues, like we said, are made of different cells. So these cells have cilia, and these cilia are important, especially those one closer to the nares or the nasal openings in the ventral aspect. So what they do is they help, those tiny hairs are very important because they help to trap in the airs, the dust coming inside our nasal cavity alongside. So they also produce the mucus, and sometimes when the mucus trapped dust, and also if there is an existing of foreign material, they trap, and then because of their proximity to the exterior, they dry, and that is why you can actually see the change in coloration of the mucus is basically because of the dust in the air that we inhale. All right. So lastly, we say that the mucus producing cells are also found in this particular nasal cavity. And we said the cells produce mucus, which helps in moistening the nasal cavity and also trapping particles. We do not want our nose to be dry. Whenever our nose dry, we tend to have this irritation, itchiness. So we want it to be moist all the time, such that they perform their complete function. One is to dissolve the smell molecules, which are also called the odorant. You need it to dissolve, and then such that you have single molecules that can easily bind to the cilia of the receptor cells that we're going to talk about, that are responsible for smell. So like we said, the nasal cavity is lined with mucous membrane. We have the concha, which are also called the trabeculate, and we've seen the superior, middle, inferior, superior, and middle. And we also see they have meatus, and we said they are important because they warm, humidify, and also filter inhaled air. So this is a real life nasal cavity, but this has been separated by the perpendicular septa, so we don't get to see the nasal cavity proper, but here we appreciate the perpendicular structure. However, if you notice an area that has been circled and it's written CN1, this area is important because it has the cribriform and the cribriform plate, that is a very thin bone found above it, and this allows for the perforation of tiny white fibers. So you can see some tiny white fibers, like web structures around that area, which was cycled as CN1. So these fibers that we see them are actually the olfactory nerve. So the olfactory nerve are nerves that combine together to give rise to the olfactory bulb, and the olfactory bulb is the first cranial nerve. Okay, so that is what it means. Then the lower aspect, we're going to talk about that, where we see V2 and also V3. We're definitely going to talk about that in later slides. So here, if you notice it, we're going to talk about the three different epithelia, and whenever we talk about epithelia, we mean it is the histology of the mucosa lining the nasal cavity. So in the most superior aspect, you can see we wrote it is written the olfactory bulb. So the olfactory bulb is a chunky area where all the collections of the nerves that left the nasal cavity is. So the bone that which they are lined and it is perforated is called the cribriform plate, and this cribriform plate is part of the ethmoidal bone. So ethmoidal bones are one of the bones that form the cranium of the skull. So ethmoidal bone combines with other bones to give rise to the cranium of the head, and then that bone at the lower aspect of it, we have a very thin bone that is perforated by tiny hairs of nerve. So that perforation is, those bones that was perforated, is called the cribriform plate. And then the nerves are important because they come to this yellow area, which is called the olfactory mucosa. So the olfactory mucosa is parts of the nasal cavity that has the olfactory epithelium or olfactory cells. So olfactory cells are the cells that are responsible for binding to the olfactants. The olfactants are the molecules of smell. So the molecules, when we smell something, it comes to the nasal cavity, and then they dissolve. The molecules dissolve with the moisture in the nasal cavity, that is that yellow area, and then these olfactant molecules tend to bind to hair follicles associated with the receptor cells. So the hair follicles associated with the receptor cells are called the cilia, and these cilia allows for the binding of these molecules. Thereafter, if they bind properly, they induce what is an action potential, and then after they induce an action potential, these action potentials in the cells will generate electric impulses, and these electric pulses are taken up by those tiny filaments that goes into the passes in the holes made in the cribriform plate, and then they combine to form what is called the olfactory bulb, and then the olfactory bulb becomes a little bit slower as it gets towards the brain, and that is becomes the olfactory tract, which we're going to talk about it later on in the next few slides. However, you can see at the top corner and right corner, you can see that is the histology of the epithelial tissue, how they look like under the microscope. And then we talk about the epithelia of the nasal cavity in the most ventral aspect. So these are made by the stratified squamous epithelia. They are smooth, and most of the time squamous epithelial cells are meant to produce hairs. So that is why we have the hairs around that particular area, and those hairs that are growing are the ones that will trap in dust and foreign particles that are coming to our nasal cavity. At the same time, these mucosa or cells produces mucus that trap on any dust that is coming. And then finally, we have the respiratory epithelium, which is located more on the posterior aspect or the middle aspect, that is the blue area. So, and then you can see that the respiratory epithelium also has its own glands that have been produced. It has several glands, and these glands are important because they produce a lot of cells that warm. So their intention is to warm the air before it goes into the trachea and then the lungs. Okay, so that is what they do. So here is an illustration to depict more of the respiratory olfactory epithelium, and then this is a magnified view. So above there you can see the olfactory bulb and olfactory tract, and then we are not seeing very clearly the olfactory nerves, but it has been magnified for us in this small box on the more right hand side, where we can actually appreciate the yellow cells. So those yellow cells are actually the olfactory epithelium, and at the bottom you can see we have supporting cells, just like we saw in the test box where you have the basal cells. So here we also have the supporting cells as well as the basal cells. So the basal cells are important because they serve as stem cells. They can differentiate either to the olfactory receptor cells, or they can differentiate to become the supporting cells. And we need this to constantly be in a cycle where we are constantly replacing those epithelial cells. This is because our nose is very open to the environment. So these cells are faced with harsh reality of maybe the cold environment or the harsh hot weather, or we are breathing in dangerous air into our system, and thereby these particular cells are prone to damage by the different environmental condition, and there it is important for us to gradually replace these lost olfactory receptor cells and also the basal cells. So the stem cells are constantly recycling themselves via mitosis, and then they undergo differentiation to either become a olfactory epithelium or a supporting cells. And we said that these olfactory epitheliums are specialized tissues that are lining the upper part of the nasal cavity. So you can see it most upper parts, in fact above the superior concha, and contains olfactory receptors, which are these yellow receptor cells in the image to detect odor molecules. So odor molecules are referred to as the odorant. And then we also see at the base of these receptor cells, you can see tiny hair cells, which are called the cilia. These tiny hair-looking cells are important because they trap the dust and also pollen and also other particles. And in this aspect, they also have the receptor binding sites for the different odorants or odor molecules. So here is an image where it depicts that particular aspect where when one perceives the smell comes into the nasal cavity, and most of them go directly to the olfactory epithelium. So the olfactory epithelium are the epithelia at the roof of the nose, okay, and that is where you can actually see some tiny fibers of the olfactory nerve. And then the olfactory nerve, when they go above, they pass the cribriform plate and then they combine in a collection network which is called the glomerulus. So this is called the olfactory glomerulus, where it is looking like a ball and a lot of fibers collect themselves and synapse to another second order neurons, and these second order neurons are either the mitral cells or the tuft cells. So these mitral cells and the tuft cells are the second order neuron cells that goes into the brain and forms so the axons form what is called the olfactory tract as they go towards the brain. So here on the right hand side, we can see the different structural layer. So in the bottom here, you can see the mucous layer. The mucous layer is where it is moist, it's being produced. So this mucus is being produced by what is referred to as the olfactory glands, or sometimes you call it the Bowman's gland. So the Bowman's gland is the one that produces the mucus, and it comes into the nasal. It becomes the olfactory mucosa. These olfactory mucosas wet the cells there, and also it helps to dissolve the smell we perceive. So the smell we perceive is made of molecules, and those molecules are called the odorants. So when they dissolve, they give rise to different particles, and then different particles, because of the chemical compositions that are now floating in that particular area, trying to bind to their exact receptors. And if they bind to their exact receptors, they can induce an action potential, and this action potential flow through these yellow cells that are found in the olfactory epithelia of the nasal cavity. And then if you follow the axons, okay, so the long lines are running from the yellow cells going upwards. So when they go upwards, you can see a thin bone there. That thin bone is called the cribriform plate. So as they flow there and they come to collect themselves in the superior aspect, which is labeled olfactory bulb, they collect themselves in a pattern which we are going to talk about later on. And these collections are formed in what is called the olfactory glomeruli. So they form like a ball-looking structure. That is why it's called a glomerulus, like a globe. So that is why I call it glomerulus. And then after they form that, they synapse with. So when they come in, they synapse with the dendrites of either the mitral nerve or the tuft nerve, which are nerves that go into the brain, forming what is called the olfactory tract. So when we magnify this particular olfactory layer, we're going to see the olfactory epithelium, as you can see it all the way, while the laminar layer is where it's mostly like a basement membrane where the stem cells are the ones being produced. So in this image, the blue cells are the basal cells, which are the ones that are becoming stem cells. They have the ability to either differentiate into the green cells, which are the olfactory epithelial cells, or rather they differentiate to give rise to these pinkish cells, and these pinkish cells are called the basal the supporting cells. And then at the base of the bottom of the green cells, you can see those cilia. So those cilia are where the tiny dotted structures, which are odorants, or other molecules bind and then induce action potential, while the longer parts of these green cells go as the olfactory nerves, and then they pass through the cribriform plate as they go upwards, but it's not shown in this image. So before we go into discuss about the sense of smell, let's understand the blood supply. So we are still talking about the anatomy. So the blood supply to the nasal area is mainly by two branches of artery. So you have the ethmoidal artery. So you have the uh, uh, and internal nasal branches of the anterior, and as well you have the lateral branches of the posterior ethmoidal arteries. So both ethmoidal arteries are coming from the ophthalmic artery. So the image above, the artery coming from internal carotid artery, it comes off to go and supply the eyeball, but we have a branch which is called the ophthalmic artery. So this ophthalmic artery is going to divide into two, where you have it's going to give rise to two branches. You have and you have anterior, and also you have lateral ethmoidal arteries. So these anterior ethmoidal artery and posterior ethmoidal artery give rise to the lateral branch, to the nasal branches. That is why you see the one on the front that goes all the way to the angular artery, the one in the most ventral aspect, and then the posterior aspect. Then the one that is more bottom here, it's called this sphenopalatine artery, and this sphenopalatine artery is a branch of the maxillary artery. So the maxillary artery is a terminal branch of the external carotid artery. So let me go back again. So the sphenopalatine artery is a branch of maxillary artery, and maxillary artery is a terminal branch, is one of the terminal branches of the external carotid artery. I hope you get that. So that is those are the main arterial supply. And then you also have another one coming from the facial artery. So the facial artery is mostly supplying the tip of the nose, and then that is where they also combine with the arteries coming from the anterior ethmoidal arteries. We also have the great palatine artery and the superior labial artery. The great palatine artery is also one of the branches of the maxillary artery, and we said the maxillary artery is a branch of the one of the terminal branches of the external carotid artery. So venous drainage follows a similar pattern, but this time around ethmoidal branches, so the branch of veins draining the nasal cavity on the anterior aspect and posterior aspect, drain into what is referred to as the cavernous sinus. So the top image on your right hand side is the base of the human skull, and then you have this venous plexus, you have these sinuses, and then the one on the medial aspect, most middle aspect, is called the cavernous sinus. So that is where the veins, the blood is draining from, and the nasal cavity, it goes into that, and then it goes through either the superior and inferior petrosal sinuses based in this image. So if it goes into the superior petrosal sinus, it drains into the transverse sinuses, but most of them will come back and drain into the sigmoid sinus, and from the sigmoid sinus it drains into the internal jugular vein. So then we've seen the one that also drains the posterior aspect of the nasal cavity, also from the sphenopalatine veins. They all drain into the pterygoid plexus, and the pterygoid plexus will also drain into the internal jugular vein, while some of them will go ascend and send branches to the cavernous sinus, and from the cavernous sinus they drain into the transverse or sigmoidal, and from there they drain into the internal jugular vein. So let's talk about the innervation. Okay, so we're not concerned about special senses here, but rather general sensory here. So the olfactory nerve, you can see, is the most area which takes up the most superior aspect, which is written as CN1, that is the cranial nerve 1. And then we also have the anterior part where we have V2 is coming from the maxillary nerve, which is the V2, we said, is the second branch of trigeminal nerve. So when we talked about the oral cavity, we saw the third branch of trigeminal nerve, which gave rise to the lingual nerve that came to assist in the sensory perception of the tongue. And then here we also have the maxillary nerve, which is the second branch of the trigeminal nerve. So V normally is a Roman number for 5, and then second branch, that is why you see a base root of 2. So it is telling us it's the second branch. And there we also, these second branches, they give off to some smaller branches as well. We have the nasopalatine, and then we have the infraorbital nerve coming from the anterior aspect and on the lower aspects of our eyes as well. So also some innervation to the nasal cavity, we also have the nasopalatine, which we just said is a branch of the maxillary and provide general sensation to the lateral walls and the septum. And then we have nasociliary nerve, which is a branch of the first part of the trigeminal nerve. So the first part of trigeminal nerve is the ophthalmic nerve, and this nasociliary is a branch of the ophthalmic nerve, and it also involves in general sensation that innervates the lateral walls as well as the septum. So also, talking about the innervation, we have the trigeminal nerve is the most common, and it also gives rise to the sensory of the axon at the skin. So that is skin sensation when we touch the skin. However, when you try to pinch the inside of your nose, the pain sensation there is taken up by the nasociliary as well as the nasopalatine fibers. Then lastly, we have also the facial nerve, which is the most nerve for facial expression. It is important because of the contribution of the nose, and also helps in the nasal cavity as it goes into the apex of the nose. So now let's go and start talking about the olfactory system. So olfactory system is very important because it's it tastes, it's function in flavor of smell detection, and the method is using the olfactory sensory neurons that convert chemical stimuli to electrical stimuli, and then it goes into the brain. So here are the different structures that are important in that olfactory area. So here you can see it's magnified, and there you can actually appreciate that the small fibers coming from the roof of the nose is called the olfactory nerve endings, and they are nerves, the ones that are going up to pass through the cribriform plates are the olfactory nerves, and then they all collect as a olfactory glomerulus, and that is the circular bulb, which is synapse with either the mitral or the tuft nerve, and those are found inside the olfactory bulb. And then when they narrow down to go towards the brain, they form what is called the olfactory tract. All right, so here we can see that the mucosa is important because it makes up the cells, the epithelial cells, and so this olfactory epithelium also releases mucus by the Bowman's capsule in that particular area. And then at the ends of the olfactory cells, we have the cilia, or the hair-like looking projections that have the receptors that will bind to the odorants or odors molecules. So this olfactory receptor cells, we said, one of the type of the cells at that particular olfactory epithelium, and they carry on the fibers. So there are fibers that are leaving the cells going upwards are the efferent fibers, and you remember this is the first point of call. So these are the first efferent fibers that goes through the cribriform plate as the olfactory nerve and synapse in the glomerulus ball with the second order neurons. So the second order neurons are either the tuft or the mitral cells, and then those axons go to the brain. Okay, so here we also see this arrangement in this particular image from the bottom aspect in the image B, where you can see the red arrow is indicating the flow of air containing the odorant molecules. And then first you can see that we have the mucus, which is being produced by the glomeruli. So you can see the olfactory gland in green, and then you can see in yellow we have the olfactory receptors, and then supporting those olfactory receptors we have the supporting cells. So these odorants, when they come here, they bind to the cilia, which is not very clear in this image, but the cilia are found inside the mucosa, and then they tend to induce action potential. Like I said earlier on, the pattern at which the olfactants bind also is unique. Just like we remember when we talked about the different textant, we talked about salt, we talked about bitterness, which are alkaloids, we talked about sour, which are and then we also talked about sugar, which are fructose, and they have the particular binding points on the taste buds. So these ones, they also have a similar way where the odorants will bind in a similar pattern with an exact receptor to actually induce action potential. And then these information are taken up by the axons, which are the afferent fibers. And because this is the first point of call, these are called the first order neurons. And then you can see the fibers, they collect, the way they collect the filaments of the olfactory nerve, the way they collect themselves is also unique. And then when they pass this cribriform plate, which is also part of the ethmoidal bone, and they form this bulb, the synapse with the second order neuron. So you can see majority of the second order neuron are mitral cells or neurons, and so they communicate or connect or synapse with the second order neuron, which are mitral nerves. We also have the 12th nerve, and then when they do that at what is called the glomerulus, and that is the ball looking structure. And then this is all the collection of this bulb looking structure is found, they form what is called the olfactory bulb, and then they narrow out as the olfactory tract going towards the brain. So this is an image where you can appreciate the cribriform plates and also the olfactory bulb. Then you can also get to see the filaments of the nerves, and then as they pass, they collect themselves, they pass through the cribriform plate, and so those are the ones that the nerve, the olfactory nerve, and then they come into the olfactory bulb to synapse in what is called the glomerulus, and then they synapse with the mitral nerve, which are the second order neuron, and then gradually they narrow out as the olfactory tract. So this is also an image to show that. So here we can actually see this is more magnified. So here you can see the cilia or olfactory cilia in the mucosa from the bottom aspect, and then you can see the pink and orange cells. The orange cells are the olfactory cells. And then don't forget, you can also see the Bowman's gland, which produces the mucus as well. So here you can see the filaments, the filaments of the receptor cells are collecting in a specific pattern, where you can see it's labeled as axons. So these are actually the axons of the olfactory nerve, and when they pass the cribriform plate, they now synapse. You can see the synapse as a glomeruli. So plural is glomeruli, singular is glomerulus, and then they form this particular synapse between themselves and the mitral nerve, which are the majority of the second order neuron. But we have some other cells which are called the tuft cells, which are also second order neurons. So this is also another image to show you that. So now you can see we are going more magnified and focused now. So you can see that neurons, all the molecules coming to bind with the cilia located inside the mucosa, and these the axons of them are the afferent axons are sent through or passes through the cribriform plate or the perforation of the cribriform plate, and then they communicate with the dendrites of the mitral nerve or the tuft nerve as a second order neuron. So you can pause it and understand the illustrations as well. So here more magnified, so you can see the steps at which the pathway starts, where we have the odorant substance or molecules. They are airborne and they're chemical molecules, and those odorants are dissolved in the mucus, and they're dissolved in the mucus, and each cell, each neuron has about 200 cilia, which is roughly almost the same as what we had in the test buds, where we are about having about 100 cells or receptor cells in a test buds. And so you have about 200 cilia per neurons, and we have about 1 million neurons in in the nasal cavity. And these odorants bind to specific receptors, as each neurons express a single type of protein receptor. So which we're going to explain this in this particular slide. So in general, we have about 400 types of receptors in the human body, but we have more in other animals such as dogs. That is why they are able to smell us even the smallest amount of odorant. So in humans, we have about 400 types of receptors, and these gives us the different types of receptors in the body, because for each neuron they have one type of receptor to bind to. So let's imagine that blue is one receptor, purple is one, orange is one receptor, green is one, all the way to 400. That is what it means. And then you can see the different color of the odorants also in the mucosa, and what they do is the ones in blue are trying to go and bind themselves to the ones that have the receptor shape. So the shape and the color is important because the molecules are trying to go and bind there. That is what you see here. So it means that one odorant can bind to several receptors, meaning that one smell has different parts. It's just like making kose. Kose is just one food, but to make kose is you have several ingredients to make the kose. But when you try to extract the kose and to a tiny bit, it means that the one kose is going to have salt, pepper. So that is what it means. So when you break them down, as let's assume is the salt, is a smell, is an so the kose is the odorant, but the molecules, the kose have made several molecules that can break down and bind to several receptor cells. I hope you understand that. So let's say that the kose, it comes in, and then let's say salt is blue, and the pepper is purple. So that is what it means here. So one odorant can bind to several receptors. That is what it means, because odorant composes of different molecules. So I hope you understand that using the analogy of kose. And so one receptors can then if you turn it another way, one receptor can bind to several odorants. Odorants compose of different. So it means that so let's assume that the salt here, we have one receptor is for salt, one receptor is for pepper, etc. Then next we came to smell puff puff. So the puff puff, the smell of the sugar in the puff puff will come and bind, look for the receptors that will bind sugar. But we know some people put salt in that puff puff, so the salt will also come and bind with the one of that the receptor of salt. So like for example, you smell a rose, and then later on you came to smell let's say something flowery, and then what happened is that, let's say receptor blue here can perceive some molecules of the rose, and later on maybe you pick a cream. What happened is that this composition, the chemical composition of the smell of the cream is going to be different. However, they have a little bit of molecules that is common between the rose and also the cream. So that is why it is said that one receptor can identify several odorants, because the odorants can be broken down as different molecules, and that is why the molecules tends to come and bind in different receptors. I hope you understand. I hope I didn't confuse you. All right. So here it's also saying that, for example, Odorants binds with their specific receptors. Each neuron expresses a single type of protein receptors. For example, an odorant blue binds with a receptor on a blue receptor, and purple odorants binds. So the molecules, so it's talking about the molecules. So purple molecule or odorants in a molecule that comes to bind with purple receptor, etc. So they have unique, and don't forget that we have about 400 types of receptors. But because of these unique way of combining their attitudes, where one receptor or one cell that contains a unique receptor can smell puff can also smell kose because of the common denominator, which is maybe the molecule of salt there. So it means that you can use one receptor to smell different and structure, but a combination of several of your receptors, which we have about 400, can actually combine to smell several type of smell. Then all axons of the blue. So next, when you pass the molecular level, you can see that when we moved towards the cribriform plate, you notice that all the ones in the blue glomeruli, they are all blue neurons. It means at the cribriform plate, what happened is that every color tends to migrate towards one side. They are all unique. Okay, so you can see all the axons of blue comes to combine together and synapse with a blue mitral nerve, and then the purple will also come together and also combine with the purple mitral nerve in a purple glomeruli. So that is how unique the pattern is. Sometimes you might end up having a lesion at the blue glomeruli, meaning that you lost this ability to smell salt in this aspect. So even though you can actually smell, sometimes you can easily smell something at not completely 100%, but because you've lost the smell of a composition of that smell or odorant, for example, where we use the salt here, where you lost the ability to smell salt, so you can actually smell a degree of the kose and a degree of the puff, but because of that combined, you'll be like, something is missing, something is missing in the smell. It's because maybe you have lost you lost or degenerated the mitral nerve involved with the blue coloration here. You can pause and rewatch it to understand what I mean here. So then what I just explained is, then all the axons of blue receptor neuron collects together to bind to the second to the dendrite of the second order neuron, which is either the mitral nerve, which are the common one, the most populated one, or the tuft nerve. This synapses is known as the blue glomeruli in the olfactory bulb. Please mind you, it is not called blue. I just use an analog to explain this presentation. So here we also see the olfactory pathway. We describe them one after the other. So the first order neurons, so you can see the colors in the image and above, where you can see lines. So you can see they are scattered, the neurons are scattered all over the olfactory epithelium. However, when they try to organize themselves, they organize themselves very well, such that when they move across the cribriform plate, all the blue ones comes to collect at the blue, all the maroons, all the green. So that is what they do. So the ones that gets the first connection with the molecule is called the first order neuron, and then their axons are the ones that synapse with the dendrites of the second order neuron, which are either the mitral or the tuft nerve, and the junction at which they do the synapses is called the olfactory glomerulus, that is one or glomeruli. And then we have in the human body, we have about 1,100 or to 1,200 glomeruli in the human body, and then we have about 50,000 mitral neurons in the human body and about 65,000 mitral tuft nerve. So the mitral nerve receives an inhibitory impulses influencing the sense of smell. So these inhibitory fibers are mostly the ones in this image are the yellow fibers, which are called the granuli cells. So the granuli cells are important because what they do is they reduce the downwards noises, such that your information going to the brain are very clean, where all the noises, which are the negative feedback, are being reduced by the glomeruli. Okay, sorry, the granular cells. So here you can also see that this is a magnified section after the mitral nerve, and then this is also what is showing us in this illustration. So the mitral neuron, like we said, receives inhibition via the glomeruli cells via a reciprocal synapses. So it means, so this one is inverted, this image is upside down as compared to what we were seeing earlier on. So what you have is the periglomeruli cells. What they do is they also control the noise between the two, or all of them, so that you do not have overlapping of information, but you want to have a clean, straightforward information that goes to the brain. All right. So inhibitory feedback from the glomeruli cells to the mitral cells are crucial for several aspects of olfactory processing. So what it does a lateral inhibition, which creates a lateral between the mitral cells that receive inputs from the different glomeruli. So that is what we see, the paraglomeruli inhibition. This sharpens the contrast between the different odor representatives, so you do not have overlapping, but rather a clean smell going in, allowing for better odor discrimination. So that is why at the same time you can smell a food cooking, you can smell something burning, you can smell a toilet at the same time, and this is because they are now sharp, they are not diluted because of these inhibitory functions of the granular cells. So it also gains control, the gaining of control. The inhibitory inputs can modulate the fiery rates of the mitral cells, helping to regulate the strength of the output signal sent to the olfactory cortex. That is just basically what I just explained. So these glomerular cells can influence the timing of the mitral cells firing, contributing to the temporal coding of odor information. And this also helps us in maybe when you are staying in a room that is having a high smell of, maybe let's say, perfume, and at a particular point you cannot even smell the perfume, but when somebody comes in at that time, because the person is freshly having no sense of the smell, it's coming in, that person will smell the perfume while you do not. It's also the same thing while you are cooking, something is burning, you that is cooking might identify the sense of burn at later time as that, compared to the person that just came newly because of the fresh impulses being sent to the brain. All right, so the olfactory pathway, we said that all the axons from the first order neuron goes in, passes the cribriform plates and synapse with the second order neuron, which are mostly the mitral nerves and the 12th nerves, and then the axons of the mitral nerve and the 12th nerve are the ones that go along as the olfactory tract, and then it's going to split into two as the bottom left image. When you notice it, you can see olfactory pedicule, which is the olfactory tract, and then it's going to split into primary olfactory cortex, and the other one is going to the opposite side. So the one going to the primary olfactory cortex is going to give branches to the anterior olfactory nucleus, olfactory tubercle, piriform cortex, and also you have some going to the ethmoidal cortex. And don't forget that some of them also go into the amygdala, and that is why you have a high sense of memory associated with smell. So the moment you smell something, you have to remember it. So this is an image where you can actually see the fibers of the olfactory tract, and then the blue coloration is the feedback coming from the brain, and that is where there is a communication between the mitral nerve and these particular feedbacks coming from the brain. So the blue one, while the red ones are the indication of the mitral and the 12th nerve that are coming in, and then you can see they are synapsing with the ones coming back from the brain. So the red one, you can see majority of them go to the amygdala, which is the amygdala is, like we said, the organ of memory here, and you can see a majority of those fibers that are coming from the and the frontal septum of the brain. They are mostly as coming as a feedback or a response coming from the brain going back to the nasal cavity, and also controlling the underground noise of smell. So here we can see the axons of the mitral nerves are then replayed to the different primary olfactory centers, and which are at the base of the frontal and inferior aspects of the temporal lobe. So the inferior aspect of temporal lobe, you can also see you have the amygdala, and also you have other structures that are related to the phonics. You also see the hippocampus, which is connected to memory, and also you can see amygdala, which is mostly associated with emotion associated. So you can see how emotions and memory are connected. So emotions are amygdala at the base of the green coloration, while the base of the purple coloration is the hippocampus, which is involved with memory. And so these are where we interpret the information of our smell. So all sensory information, be it spatial or other general sensory, goes to the hypothalamus, to what is an organ referred to as the thalamus. If you remember when we talked about the deep gray matter. However, this one is the only sensory information is the only one that does not go directly to the thalamus, but rather goes straight to its own primary cortex. So we said that the primary cortex include the anterior olfactory nucleus, olfactory tubercle, and then we also have the piniform cortex, amygdala, and ethnoanal cortex, not correct, but rather cortex. And so you can see these are the locations of these particular structures, and you can see them, they're not very far away from the olfactory bulb. And so here is another image. After the primary areas, they now move to the secondary areas, which are mostly in the green color. So the green color is the secondary areas, which is the insula and also the olfactory frontal cortex. That is what it means by OFC. So here is also another image that you can pause and also study with the direction of the flow of the axons, where a lot of them go to the primary cortex, which are shown in this image. Later on, they go into either the insula or also the olfactory frontal cortex. So we also said that if this is an image which we talked about taste, and you remember the taste also follows a similar pathway, which is also associated with amygdala and hippocampus, and definitely know that these particular two senses are going to interact at either these points as well. And so this is a slide we discussed about in the earlier class, where we said simply we can just say taste and smell interact at amygdala and also the orbital frontal cortex. However, it is not just like that. It's a very complex communication or interaction between them, where they can also communicate at different points of the pathways, where you also get to see the salivary nucleus in the medulla oblongata. You can see also the ventro-posterior medial nucleus of thalamus, also in the insula. So all these are potential sites of interaction between the fibers of taste and smell, and thereby taste is being interfered by smell. Apart from the physical anatomy or structure, where we have the communication between the oropharynx and nasopharynx, or the movements of the the testants from the nasal area also to the oral cavity. So these are potential clinical application where we need to, because of the exposure of our nasal cavity to the exterior, we need to frequently replace this respiratory epithelial cells as well as the supporting cells. That is why the basal cells continue to differentiate and also replicate. However, if there is a prolonged injury to that we could have temporary or permanent types of anosmia. Anosmia is the ability of the partial or complete loss of smell, and these can be due to age. So gradually as we age, we tend to lose our sense of smell. And you have head trauma, where you have the nasal prolapse, where you have a non-cancerous growths, viral infections such as cold, flu, and COVID-19. During COVID-19, a lot of people lost their sense of taste or smell. And also when you have the sinus infection, you remember we said we have the nasal sinuses surrounding the nasal cavity, and they discharge their fluid into the nasal cavity, so thereby they can also interfere with the activity or functions of the epithelial structures there. Early insomnia is an early sign of several neurodegenerative diseases, including Parkinson's and Alzheimer's. And sometimes we have medication. When you take some certain medication, they tend to interact or give you partial loss of smell. So we also loss of smell can affect the sense of taste, like we've said. And person with epilepsy have hallucination of disagreeable odors. So sometimes they hallucinate even a sense the smell of something. All right, so thank you very much. If you have any question or any comments, you can drop it in the comment section. I'll see you in the next class. Bye.